Hello, everyone. I am Assistant Professor Dr. Habibe Yilmaz from Trakya University, Faculty of Pharmacy, Department of Pharmaceutical Biotechnology. I'm going to present to you synthesis and characterization of a novel second-generation platinum derivative and its molecular interaction analysis via surface plasmon resonance. Today we are going to talk about the background of the study, the objectives, the experimental section, the results, and the discussion. The role of epigenetic mechanisms in cancer treatments has been investigated in recent years, especially research on histone deacetylases has gained momentum. Histone deacetylases removes acetyl groups from amino acids on histone and non-histone proteins and classified into four classes. Histone deacetylase inhibitors induce hyperacetylation of histone and non-histone proteins, which leads to the inhibition of cell cycle progression and induction of apoptosis. Since the importance of histone deacetylase as pharmaceutical targets was emerged, many inhibitors have been developed and research is still ongoing. GLI1 is a transcription factor that is involved in cell phase as well and belongs to zinc finger protein family. Recently, a study showed that histone deacetylase 6 is significantly associated with splitting resistance in non-small cell lung cancer, and it is a potential novel therapeutic target for platinum refractory non-small cell lung cancer. It is located both in the nucleus and the cytoplasm, belongs to the 2B class, and is dependent to zinc ion. Both histone deacetylase 6 and GLI1 has zinc finger protein domain. Zinc and benzoic acid derivatives in zinc finger protein region form a chelate with a bidene-based structure. There are also studies showing that platinum derivatives interact with zinc finger proteins, especially with C2H2 domains. Since cisplatin and histone deacetylase inhibitors such as 4-hydroxybenzoic acids are known to interact with histone deacetylases, many combination studies have been performed and showed promising results. However, there are no previous reports in the literature on the anti-cancer effects of a platinum compound containing histone deacetylase inhibitor, which is known to avoid cisplatin resistance. Our objective can therefore be restated as to conjugate 4-hydroxybenzoic acid platinum to obtain a novel platinum derivative that does not trigger cisplatin resistance and evaluate its interaction with zinc finger proteins such as histone deacetylase 6 and GLI1. Synthesis completed in 3 steps. At first step, silver nitrate solution was added to 4-hydroxybenzoic acid and left to react for 2 hours at room temperature. In second step, 60 mg of cisplatin was suspended in potassium iodide solution and allowed to react for 1 hour at 30 Celsius degree, and at third step, both solutions were mixed and allowed to react for another 18 hours at 50 Celsius degrees. Formed silver chloride precipitates were removed by centrifugation and the supernatant was dried. The dried sample was dissolved in DMF and purified with a colonic chromatography containing silica gel 60 resin. The collected fractions were dried and analyzed with TLC, UV visible spectrophotometry, HPLC, and both proton and carbon NMRs for physical-chemical analysis. For biological analysis, molecular interaction was achieved with SPR instruments of Bio-Rad. The chip has 6 crisscrossing channels which allows to assess 36 interactions at a time. I used NTA chip to immobilize his stacked histone deacetylase 6 and GLI1 proteins. Chip was initialized with glycerol, conditioned with STS, sodium hydroxide and hydrochloric acid, cleaned with EXA, and activated with nickel sulfate solution. Varying concentration of histone deacetylase 6 and GLI1 flowed in vertical direction. Then chip surface was cleaned with running buffer, STS, and another cycle of running buffer. Before flowing over the analytes, chip reconditioned with STS, sodium hydroxide, and hydrochloric acid. Afterwards, varying concentrations of cis-4-hydroxyphenylplatinum-2-diamine flowed over in horizontal direction. Regeneration was achieved without losing of ligand with 25 millimolar sodium hydroxide, 15 millimolar hydrochloric acid, and 20 millimolar ecta. As can be seen here, a blue compound obtained. Its UV visible spectrum scans revealed a different scan profile. TLC results showed that synthesis was successful since different RF values obtained from initial compounds. To calculate the reaction yield, underreacted cystatin was measured spectrophotometrically, and the overall reaction yield was calculated as 62.7%. As can be seen from HPLC results, there are two peaks at 3.5 and 8.2 minutes, which belongs to the novel compound and 4-hydroxybenzoic acid respectively. The amino group of cisplatin and hydroxy group of 4-hydroxybenzoic acid overlapped at that region in novel compound. Also, carbonyl signals of 4-hydroxybenzoic acid observed in this region, and carbon-carbon double bonds of aromatic ring in this region shifted to here in novel compounds. The platinum chloride signal was not detected in novel compounds since chloride is displaced with 4-hydroxybenzoic acid. Hydroxyproton bound to aryl can be seen at 7.8 ppm, and for protons bound to aryl structure can be seen at 6.7 ppm. Primary amine proton was detected at 4 ppm, as can be seen here, and this belongs to the residual DMSO signal. In novel compound, there are 4 different types of carbon in terms of carbon NMR. Carbons of aromatic ring gave signals at 115 and 131 ppm. Carbonyl group gave signal at 161 ppm, as can be seen in here, and carbon that is neighbor to hydroxy group gave signal at 169 ppm, as can be seen in here. In order to obtain significant kinetic results, target Rmax values during immobilization must be achieved, which is between 50 and 100 response units. By using this equation, expected ligand immobilization yield calculated as can be seen in here for both ligands. Based on immobilization results, the target immobilization values achieved for kinetic analysis for all channels in vertical direction. During interaction analysis of novel compound with its ligands, affinity constant, association constant, dissociation constant, and Rmax values was calculated by the instrument's software. As can be seen, Rmax values achieved for both ligands. Affinity constant of novel compound to histone deacetylase 6 was 12.1 micromolar and 24.1 micromolar for Gli and Gli1. A fast dissociation based on Kd value and fast association based on Ka value was observed, and as a conclusion, synthesis completed in 3 steps and purified from silica gel 60 column. All analyses revealed that compounds synthesized successfully. The new compound binds fast and dissociates slowly to both ligands, which means it will interact rapidly with its target and allow enough time to show pharmacological effect. The presence of affinity indicated by SPR revealed that the compound's affinity for zinc finger proteins. This indicated that zinc finger proteins such as histone deacetylase 6 and Gli1 could be targets of this compound. In the light of the obtained data, it is seen that it would be beneficial to conduct further research on the interaction of the new synthesized compound with the relevant epigenetic mechanisms and metabolic pathways. I would like to acknowledge Trinity University, to Presidential Strategy and Budget Department, and ARGEFAR, because synthesis part was performed under my dissertation thesis and under supervision of Professor Dr. Şenay Hamarat Şanlar and financially supported by Ege Üniversity Scientific Research Projects Office. On the other hand, the molecular interaction part was performed independently from the thesis and financially supported by the Presidential Strategy and Budget Department. I'd like to thank to Ege Üniversity ARGEFAR for opening up their lab to me for SPR analysis, and thank you for your valuable time.